Hello, my name is Jan Maita, and I'm a leader of a team that worked in over 50 projects in which we are applying AI to multi-omics data. And in this talk, I would like to go with you through the road from the data chaos to drug discovery. But I would like to also start this presentation with a question. And I believe this question is familiar to a lot of people in this room, because it's quite known that majority of our drugs are failing at some point in drug discovery and development. But I would also like to put this question in somehow different perspective, because right now we are generating more data than ever. We are having this omics revolution, imaging revolution, and so on, and yet it's 3 times more expensive to make this drug going through all those phases than it used to be 20 years ago. And I believe this is because data generation is something that adds you actually more work rather than information itself. To get those insights, you need to process this data, and this data processing is this crucial step on the road from data to insights. And I know this, and because I have seen those stories that we are bringing our customers from data mess to actually navigating their drug discovery and development processes with informed decisions from biologically aware AI. And this kind of multi-omics journey is something that I've seen in a number of cases, because I have seen companies that we are able to help with getting their target and molecule nomination being improved from being a 3-month process to 2-day iteration. I have seen a company that was going through high throughput screening, and they were doing around thousands of assays, and we were able to slash it down to 50 assays, getting the same information. It also results in reduction of majority of costs, and also it enables you to keep your process as it is, with improving only crucial parts. But before we can go to use cases describing how we are able to benefit in the drug discovery process from the data, let's start from what is the bedrock of this data-driven insight generation. And the fundamental part here is building data products. Because when you take a look at this slide on the left, you have different data like genomics, transcriptomics, imaging, and so on. Usual suspects, you know them. It's like a backbone of the modern scientific research. But those of you who work with this data probably already know that when we have this metaphor, that data is new oil, your engine is actually running on fuel. You need to extract fuel from this oil. This is what you also need to do on the road to insights. You need to extract data products. And those data products are those intermediate entities extracted from the data on the road to the data analysis. And while it's quite straightforward when we are talking about single omics, that you are processing your genomics data, for example, to get variants or mutations. When it comes to data, to multi-omics data or multimodal data, it's much less simple, because you need to integrate different data types and different layers of informations. And this is what is embedded into those data products. And they are very fundamental for each data-driven process, because it enables you to follow the FAIR principles. It breaks down silo in the organization. Those data products are usually AI ready. And also, what is very important, those data products are built with business, or in our case, mostly scientific question in mind. So they're built for purpose, not just because you have data that needs to be processed. And when we are thinking about bringing different data modalities into one data product, there is this term data harmonization, kind of umbrella term for different approaches to make one data product out of different data sources. And the first way of thinking about data harmonization is thinking about different data domains that we are bringing together, biological domains. Like, you have different organisms, obviously, but you have also different information layers like genomics, transcriptomics, proteomics, and so on. So this is the first, probably most straightforward approach to harmonization of data. But when you think about those different data domains, those data sets like VCFs or expression matrix, they are essentially tables, very easy to work with. data format. This is something that maybe from the biological point of view may be challenging, but from technical view is straightforward. But when we are thinking about adding more data that is currently on the rise, like imaging, when you are visualizing tissues, or when you are doing phenotype profiling, when you are adding structural information, or later in clinical stage when you are also adding unstructured medical records from clinical trials, this requires another approach. This approach is called multimodal fusion that enables you to get and link information from different data types. And speaking about linking information from different sources, there is also a third layer here on the road to building data products, the data universe. Data universe, which means that what you are doing in your research project is probably not the only project in the area, in the therapeutic area, for example, and somebody already generated some data in this case. So what we are doing is that we are enhancing repositories for our customers based on their data and also proprietary data from our partners or the publicly available ones. And well, this conference is 2025, so we cannot forget also about the LLM GPT revolution that we are seeing everywhere right now. And this is something that, well, it's believed that at some point the model, the foundation model, can replace the data. Because those of you who worked in the project which involved generation of a lot of data to be able to get insights with machine learning or other data greedy approaches know how painful it is to get the samples, get the data. And so I worked in a few such projects, and actually I would like to avoid it in the future. And this is what is the promise of foundation model, that we have all this information encoded into this model, and we are able to move from generating vast amount of data to very precise generation of only relevant data that is very, very important for your particular case. And this enables us to go through something that can be called AI lab feedback loop, that you are taking your foundation model, you are adding your relevant data that comes from your lab, you are generating new insights that enables you to improve your next experiment, and you are adding this next experiment data to the foundation model. You are fine-tuning it iteratively, or you are adding it with some RAG infrastructure, it depends. And you are able to improve iteration after iteration. And those foundation models, they are coming from different areas of biology, because probably a lot of people already heard about AlphaFold. It got Nobel Prize. It was designed for structural understanding of proteins, but right now we are on the rise of different foundation models, like, for example, STGPT for single cell research, EVO2 for genomics, or recently Google announced their model, TXGenMA. So this is a very promising area of optimizing this data products building. And just to recap this part, when you take a look at this scheme, you can see how data products are central to this process. That on the left you have this data generation and this mixer from different modalities, different domains, and also different data sources. And on the right you are having what is very crucial and fundamental to the drug discovery and development process, because this is where actually insights are being generated, and they are enhancing each of those steps. And now we can go through a few use cases from the project that we already did in these areas, and we can start with the target identification, where every drug program starts. This step is very fundamental. You probably know this, that the risk of nominating the suboptimal target is something that will propagate for the rest of the drug discovery journey. And at this stage, we want to make sure that what is selected is a very proper one. And here you can see a very simplified architecture of what we are using, what we are calling a multitask model learning, which enables us to use, like in this case, we used omics data, imaging data, and information about molecules used for screening. And via this deep learning architecture, it enables us to nominate a hypothesis about what may be the proper target for this case. But when you take a look at this image, probably a lot of you think what we already heard from from a lot of people from the lab, from biologists, that this is kind of deep learning black box, that we cannot understand actually where these targets are coming from, and this makes us less convinced, less likely to believe into what we are seeing. And this is why at this stage we are not only doing target identification, but also target validation, which means that we are adding a lot of biological reasoning to this process. On the left, we have a very technical, machine learning, deep learning heavy step of joining different data types and selecting important features. What you have on the right is the process of validation data with regard to one exemplary biological process. In this case, we are using synthetic lethality phenomenon. Synthetic lethality, which means that when you are having a pair of genes, one of these genes can be deactivated, for example, by mutation, but the cell is still viable. It may be broken, but viable. But then you have the second gene that if you deactivate both of those genes, the cell become dead, and the disease, for example, the tumor stops to grow. And this is the algorithm for verification whether pair of genes may be synthetically lethal. We published it in 2023, and it's also available via application, slidevip.app. You can go there, you can enter your pair of genes, and based on data from TCGA, CCLE, and other similar resources, you can take a look and understand how likely to be synthetically lethal your genes are. And those target identification areas, this primarily works with omics data, with data that comes from different biological domains. But a nice example of multimodal fusion and integrating different data types on a technical level is Ardigen platform that works actually in the further stage of this drug discovery journey, where we are looking for the right molecule. On the road to this, we are using cell painting data. We are integrating those images also with information about ligands and with omics information on cells that are to be used. And using this platform, after this multimodal fusion, we are, first of all, obviously, generating information about the potential hits, but this is not everything that we can get, because the molecule that can be effective is only one thing, but to describe other properties of the molecule, like for example toxicity, and predict this toxicity early in the process when you are nominating the molecule, this is another level of understanding of your molecule, whether it's a right move to go with this one or to screen longer, screen deeper your library. So this is what Ardigen is about finding the right molecule and also understanding how this molecule may behave, what may be the mode of action, what are the PK/PD parameters, what may be the toxicity, and so on, to know it as early as possible. And there is also other very important things on the road between drug discovery and translating it into the clinical trials. And a very important part is about choosing the right model, because we see customers coming to us one after another and asking us for help in the selection of the right model, not only because humans are much more complex than cells, but also because those cells or mice, they are different on a genomic or other levels. Even the microbiome sometimes is described as a factor of importance in terms of selection of the right model. And once again, here at this stage, we have in our portfolio this project where we're using data from different sources like TCGA, CCLA, and so integrating this with information about patients, how the cohort, the real cohort looks like, to find those models that are most resembling with what the company we were working for is seeing in the preclinical phase of this study. And speaking about trials and clinical trials, there is another case here in which the company approached us before entering phase 1. So they already had the drug and they wanted to make sure that the clinical trial will be as smooth as possible. And they asked us what we can do to help them to make sure that they will be enrolling the right patients. And we came up with this idea that we can develop for them a sensitivity biomarker that will predict how likely patients are to respond to the treatment. We used information about cell lines, we used preclinical information from their internal data sets, and we also used information publicly available about how cohorts look like in this disease. And we were able to identify molecular pathways associated with sensitivity treatment. And this enabled us and our customer to stratify patients, to make those segments in their population and enroll those who are likely to respond. And what is very important about applying AI to data in the drug discovery and development process is that not everybody, obviously, in this process are data scientists or bioinformaticians, and not everybody wants to code their way through this data to get insights. And this is why we came up with this idea of AI workbenches, which are essentially applications that beneath you have some very, very robust model. Like here in this slide, you can see a screenshot from a Phenate application. Like 2 slides ago, I was showing this hit ID model that was doing multimodal fusion to select the right molecule. But this model can be operated with a very simplified interface where you're loading your imaging data, you're also loading some omics information about the ligands, and the scientist in the lab is able to run this analysis on their own without waiting for bioinformatics team, without getting this burden, getting those insights like with a self-service way. And this gives those companies a faster turnaround when it comes to validating some ideas, and also it allows those scientists not to be frustrated with all those challenges in internal communication, and also with very limited bandwidth. And what we can do when we are having all those ideas, all those both data, product generation, knowledge, and also insights from those case studies on the road ahead. And this is the pattern that I see from project to project when we are working with data and with our customers. There are 5 steps that we can summarize as a kind of AI-enhanced data strategy, something that can be applied to a lot of different drug discovery processes. And the first one is to assess data maturity, because we are actually approached by company at different levels of data maturity. There are companies that are very well established when it comes to the way they are generating data, and storing it, managing it, and building those data products. There are companies that are actually earlier in this stage. They are obviously generating some data or ordering data at CROs, but they are not having maybe resources for robust data management. The second stage is mapping existing process of drug discovery and development at this company, to understand which stages at this process are actually the most time and budget consuming. Because when you, for example, think about this hit identification case, well, they were having a process of drug discovery, but they understood that this process of nomination of a molecule from a library is very time consuming and very, very costly, and they asked us to apply AI to actually this part. The third stage here is what we already called AI lab loop. So to close this loop with some infrastructure, with some software, so that when you are having your lab generating data and you are having this AI, they are integrated. The data that you generate in the lab is used to feed your model, to make it better, and also what model is predicting to happen. Those insights that are being generated are navigating your efforts in the lab. The fourth stage is applying some impact metrics so that you are able to measure the process. You know whether your model is going in the right direction or not. This is very important, especially because we want this AI lab loop to be as automatic and as unsupervised as possible, so that those metrics can be included into the software infrastructure here. And last, but probably the most important part here, is to be open to refining your experiments, your strategy upon this feedback, so that when you are having this model generating insights based on your data, you are open to navigating next experiments upon what you are seeing in those insights. And this is actually what we are doing at Ardigen. We are applying AI to drug discovery process to make it more fail-safe. We already did over 500 projects in this area with over 100 customers, including 16 of biggest pharmaceutical companies in the world. We are around 150 people with a lot of PhDs from computer sciences, bioinformatics, biology. We are actually right now 10 years in the market, so we were here far before ChatGPT, and we are still developing ourselves. We are publishing internally and also with our customers or with our partners, both scientific and technological. And if you want to learn more about what we are doing and how we can help your drug discovery program, you can meet us on the booth that is in the very middle of the room, the one with the big shiny wall, and we are happy to talk with you. Thank you.